Well, thank you all for coming. It's a very kind introduction. And I think it's sort of appropriate that this is a hybrid seminar format because it's sort of a hybrid seminar topic. As you can tell from my background, I sort of started in bioengineering, did some work on using GPCRs as sensors and microfluidic devices for GPCRs as environmental sensors. And then went into consulting for a couple of years, basically sold out, and then returned to academia to work on policy and to run academic research centers. So during that time running both the Cyber Center and then the Wild Neuro Hub, I've tried to keep in touch with research and to keep my own kind of research going on the side. And so that's what informs the three projects that I'll show you here. But most of the reason why I got in touch with Bill and why I'm really interested in Middlebury is because I would love to kind of get back into doing research more, approaching full time, and to work with many of you. I really see this program as one of the kind of intellectual stars of this field, driven a lot by Ray. I think you'll see that Ray's kind of ideas have informed what I'm interested in, and obviously Ray has done much better work than what I'm showing here. Nothing I'm showing is going to be sort of a bombshell. I'm not going to be revealing any kind of state secrets. These are mostly in-process projects and ideas that I've had. So the first thing that I wanted to talk about is I'm kind of framing my talk around cyber biosecurity, mostly because this unites kind of two of my interests, cybersecurity and biosecurity. On the left is a framework that's sort of commonly used to think about what is included in cyber biosecurity, so anything from cyber threats and system integrity, what we would think of as traditional cybersecurity topics, to security of patient portals, security of patient data and of genetic treatments, digital medicine, tracking pathogens digitally. These are all sort of interesting components, and I kind of simplify them into, on the right, a framework that involves 4 tasks that I think come up a lot in cyber biosecurity topics: detecting and identifying outbreaks, whether they be natural or purposeful, and detecting unsafe conditions for research, monitoring research and ensuring that actors who have access to tools and data are using them safely, protecting and securing systems and individuals from risk, and then remediating, either cleaning up an outbreak or restoring compromised systems and healing the sick. So the 3 projects that I'll tell you about, in terms of monitoring, I'm looking at monitoring publications and downloads that can be indicators of sort of baseline research activity, where we can feed these into a model that will inform where research may be absent where we would expect it, or where research may be going on in greater sort of quantities than we would expect from the number of researchers or the funding that's available. I'm looking at another project at a cybersecurity assessment of biofacilities, partially driven by the idea that cybersecurity has kind of improved in ways that I think is not reflected in practice in both medical facilities and in manufacturing facilities, and hoping that we can move toward a kind of simple device security assessment that we could ask labs and manufacturing facilities to provide to ensure that they're being protected from cyber threats that could lead to leaks. And then the third project, and also in the monitoring domain, is on a proposal for a partially homomorphic DNA synthesis screening cloud. So the idea that DNA synthesis providers currently kind of agree amongst themselves to be part of this consortium, where they will screen the sequences they're being asked to synthesize against a library of known harmful pathogens. There are a lot of problems with this. One, obviously, is that this is a voluntary mechanism that not every synthesis company has to adhere to. And the other is that there are multiple companies, and currently if they share sequences, or if they share the results of this screening at all, or if they use the same screening tools, that is also voluntary and not entirely coherent. So I'll show you this proposal for a way to be able to allow them to share sequences and to share screening results while retaining fully encrypted control of that data. So the 2 kind of threat models that I'm thinking of in cybersecurity, we think about widely used software that has pre-existing sort of naive vulnerabilities, has 0 days. Vulnerabilities are discovered by malicious actors and either exploited directly, for example, through phishing or credential stealing to exfiltrate data, or breached through negligence, which is much more common than the exploitation of 0 days, but somewhat less likely to cause harm. And there's kind of a focus on flashy and complicated attacks, even though most attacks leverage basic vulnerabilities. The vast majority of cybersecurity incidents that we see are exploiting a vulnerability that has already been published, and in most cases a vulnerability for which a patch already exists, and it's just that kind of compliance with remediation of devices lags identification of device vulnerabilities. And we can compare that to biosecurity, where we have kind of widely used wetware that has also pre-existing vulnerabilities, malicious engineers maybe use an engineered or pre-existing pathogen to cause disease or harm, and they can weaken or circumvent either existing protective measures, the immune system, where they can introduce a pathogen into a common supply or into a crowd. Breach through negligence is common, and there's kind of a focus on more harmful diseases, but most attacks fail, as opposed to cybersecurity, where I think we see that most attacks succeed, and that your personal data has likely been breached multiple times, even in the past few years. So what informs kind of some of this first project is the Bioweapons Convention and the introduction of confidence building measures. So Bioweapons Convention involves semi-annual, recently not quite semi-annual, but an exchange of information in order to prevent or reduce the occurrence of ambiguities, doubts, and suspicions. And the confidence building measures were first agreed upon at the 1986 Second Review Conference and defined in 1987, and these require annual declarations on exchange of data at research centers and laboratories, exchange of information on outbreaks, encouragement of the publication of results of biological research, and the active promotion of contacts between scientists engaged in biological research. These are the things that have to be reported on through the confidence building measures, and the states parties, of which there are currently 183, are asked to implement the measures in the BWC and asked to send in a report on the CBM. So this is a set of 6 forms that they're to complete and submit by April 15th. In 1991, there were some additions and sort of improvements to the confidence building measures. These were developed and reorganized into a new set of 7. So for example, some of the changes, CBM-A changed the declaration of high containment facilities or declaration of national defense activities. CBM-E required the declaration of legislation and regulations and other measures that will implement the Weapons Convention. CBM returns have been trending upwards, but not 100%. So there were 90 of them submitted in 2021, 36% of them made public. You can go and read them. They are not a particularly onerous reporting mechanism, and yet we see slightly over 50% return on them. So one of the kind of difficulties or frustrations with the BWC is that it is underfunded in the sense that a lot of the state, well, not a lot, but some of the states parties don't pay the required dues, and the implementation committee for the BWC is 3 people. There's no, as you know, there's no verification part of the BWC, mostly due to the US disagreeing with the site visits part of the verification proposal. And so really, kind of all that we have, I think, in order to make sure that that states parties are conforming to the BWC are these confidence building measures. So it's nice to see that that is trending upwards. We would like to see more increased compliance with it. And in the 2018 meeting on the BWC, we saw some working groups and discussion groups formed around sort of topics of disagreement or topics of discussion, including strengthening cooperation, reviewing developments in science and technology and genome editing, strengthening implementation, assistance and preparedness, and institutional strengthening of the convention. So the projects that I'll talk about, I'm not sure, well, there's some highlighting here you probably can't see here, but the projects I'll talk about are mostly to do with the third topics in terms of exchanges of technological capability, which I'm going to talk about in the section on the cybersecurity assessment, reviewing developments in technology and strengthening national implementation, which I'll talk about in projects 1 and 3. So if we think about, these are all the things that the CBMs require states parties to provide information on, which of these can we easily track through open source information? Or maybe not easily, but which of these could we possibly track through open source information? So we can track research centers and laboratories, their existence, their footprint, 3G satellite data. We can track national biological defense research and development programs insofar as they're reported on or insofar as the funding for them is publicly available. Outbreaks we can track to a greater or lesser extent. There's been some interesting work on models to predict outbreaks of something like African swine fever that's endemic in Russia, and being able to predict it rather well through just weather data and tracking the local populations of wild boar to see where they come into contact with farmed populations of boar. So I think there are a lot of kind of interesting ideas there in terms of where we can track outbreaks. But publication of research results we can certainly track. Promotion of contacts I think is one of the most difficult. I am meeting with LinkedIn next week to see if there's a way that they can give me access to their data so that I can look at Russian researchers, because you used to be able to scrape LinkedIn, and then they cut that off, and now they don't want you to scrape LinkedIn. So now that's no longer open source, and I'm trying to get it through closed source methods. But so that I think would be a good way of tracking promotion of contacts, as well as publications of conference proceedings, talks given. Legislation we can certainly track. Past offensive or defensive programs we have some information on, but again, only where kind of admitted to. And then vaccine production facilities, we can more or less track, but I'm, I guess, less interested in that. So in terms of going back to the middle one, publication of research results, what can we look at? So publications, conference attendance, and travel. We can look at GenBank uploads. We can look at what kind of proteins or what sequences researchers are working on to indicate the scope and volume of scientific activity by country and by topic. We can look at sources like bioRxiv, Google Scholar, PubMed, Scopus to collect publications and author affiliation data. And then other potential sources like for bioinformatics researchers, GitHub repositories, again, LinkedIn connections, Sci-Hub traffic if we're able to see that. And the motivation here comes from kind of what I think of as insider threat modeling applied to non-compliance. So the idea that we're using kind of this second column here of event detection and behavioral variability analysis, monitoring divergence from normal behavior, and looking at how bad behaviors can be extrapolated out from a source of data where we are kind of modeling positive behavior. Or another way of looking at it is kind of these three ways of thinking about this in terms of, are you either modeling positive behaviors, are you using anomaly-based detection models to model legitimate behavior and identify the degree to which observed behavior deviates from that, or are you modeling negative behavior, looking at misuse-based or signature-based detection models to model what you think of as malicious behavior, identify again the degree to which observed behavior conforms to that, or are you using kind of a hybrid approach of outlier detection in cases where neither legitimate nor illegitimate behavior can be modeled? So if you have a small number of use cases or behavior that varies greatly, can you just measure deviations in internal consistency? And these sort of come from insider threat detection within a cybersecurity frameworks. That's what we think of either anomaly-based detection or misuse-based detection coming from. So the first thing that we want to think about in terms of what sort of signal are we looking for is how many people are we trying to find? So something you're probably familiar with from a non-proliferation review, which countries have known or suspected programs, and at least historically, how big were they? So like, how big is the signal that we're trying to identify here? Are we looking for a handful of researchers, or are we looking for many thousands of researchers? Based on this data, program sizes varied over the Soviet Union's historical 60,000 people. The US maybe employed 6,000 people over the course of their program. South Africa maybe employed 5 or 6 people. So this can inform the level of signal that we're looking for. And we could be looking for quite a small population. I mean, if you think about 6,500 white-collar crime prosecutions in the US per year out of 88.5 million white-collar workers, even if we assume only a 10% clearance rate or something, we're still looking for a very small population within the overall researcher population. So here's some work that I did with Elsevier, which is a publisher, and they allowed us, it is open source data, you can scrape this, but they kind of allowed us access to their data internally as well. Initially, we wanted to look at modeling positive behavior. So where do we expect to find activity? So which pathogens are under and overrepresented in the literature? Are there candidates with understudied virulence potential, or are there candidates maybe with sequence variants that are not in GenBank and so not covered by the screening protocol? And so those are kinds of things that we would expect to find in the middle of this tail. So as you might expect, when we look at publications by topic area, the US has the most publications on every disease except for the ones listed, every disease we look there. And then we kind of pulled out some outliers as well, where we can see that, Australia publishes the most on Hendra, for example, which makes sense because Hendra was initially discovered in Australia. And so it's kind of anywhere where you would expect to have a natively occurring or endemic population that have this disease, you're going to have more publications. And then what we're looking at here is just the number of studies by pathogen. And the pathogens here were ranked in terms of their presence on the US's like A, B, C threat kind of classifications, which is why I guess you might expect it to be by a number of publications, but it's not. So we're looking at kind of worse pathogens to the left, not as bad pathogens to the right, I guess. And then here's just the number of countries where these pathogens are detected. So the number of countries that have these. So if on the left you have something like HIV or HPV that is detected in almost all countries, and then over here you would have something like Hendra or one of the flu variants that are detected in relatively fewer countries. So there's an almost a linear relationship here, and I think again what's most interesting to look at going forward are the diseases in the middle, where they are found in a wide variety of countries, but they're relatively understudied. So that to me would indicate that, again, they might have sequence variants that are not in GenBank, there might be virulence potential that's understudied, and so these might be good targets both to track what research is being done with them, and also, if I were interested in increasing the virulence of one of these pathogens, I might focus on one of the ones where I would think, well, the sequence variants are not being scanned for as harshly. So we can look at how has research activity and investment changed over time. So this is the same data source, number of studies, and here we're just looking at some of the kind of big players, so anthrax, plague, Ebola, H5N1, H1N1 in blue, and then SARS, and obviously we stopped looking prior to COVID. COVID kind of messes up a lot of this data, so you'll see a lot of the studies, I stopped at 2018 or 2019 because I really don't know how to analyze that. A lot of labs shut down during COVID. I think it's just going to be kind of a lacuna in what we can look at in terms of research productivity. But so that's why this ends at 2017. So the trajectories differ. In some cases, you can see research productivity spike because there was an outbreak or because there was an event in the news. I think that kind of makes sense. And we tried to categorize the trends in terms of number of published studies in this order. So whether there's just kind of sustained growth, over time, relatively more and more people are interested in this. And this is something like dengue, like rabies, pneumonia, a plateau followed by an increase followed by a plateau, and so on. So we can classify different levels of interest, and this can also help us then predict what we expect interest to be going forward. And then I think it's also interesting to look at trajectories where we don't see a trend, so category D, where there's kind of an irregular trend of interest, monkeypox, Nipah. So these are relatively less studied, but where we can't tie the increases and decreases to external events. So that's something that might be interesting to look at further. Some of this is just to show kind of the scope of what we're looking at. So these are article counts as a point in time, and so we have the countrywide distribution here, the number of countries in which the disease is found, and then the number of articles, and these are separated out by, so viruses over here, bacteria, and so on, to show what we were kind of looking at and how they were grouped in the study. And then we also looked at article counts by country as a point in time. So we can then start to monitor changes in these distributions. This is how many articles reference these pathogens by country. And then we can feed this back into the model and look at monitoring changes in these distributions over time and whether these correlate with changes in funding, changes in defense priorities, as stated publicly by the country, maybe changes in dual use research or changes in research capabilities, researcher migrations of someone who publishes a lot on Hendra, leaves Australia but still wants to work on Hendra, maybe we see a spike there. So this was kind of intended to feed back into the model so that we can then think about what other sources do we want to look at? Do we want to look at funding? Do we want to look at researcher migration and so on? Then we also thought about what if looking just at publications is too blunt? Someone can be doing research and not publishing on it. So then we also wanted to measure downloads. So using both Scopus and ScienceDirect, we identified IP addresses for article downloads. The highest traffic hubs are shown on the map, and those generally correspond to universities, as you might expect. We were thinking initially of using the highest traffic hubs as a kind of modulus so that we could take the actual data and remove traffic from those hubs. I think that actually doesn't work because, as it turns out, there are just so few downloads of most articles. In the next slide, I'll tell you kind of how we initially started doing this with Elsevier and what we were looking for, but we ended up just reporting actual downloads and not modulating them by that. Another thing we ended up having to do for the data is track how IPv6 addresses correspond to IPv4 addresses, because there was that change, and so the same two different addresses correspond to the same address. It's just that the server had to change because of that configuration. Other caveats I want to point out to this method are that articles can be downloaded from sources other than Scopus or ScienceDirect that track downloads from the actual publication website. They could be downloaded through a VPN. They could be downloaded through a resource like SciHub. They could be read in full on the site of an open source journal rather than downloaded. So we're tracking downloads. That's not a perfect proxy for reading or interest in articles. But we were interested in determining whether we could track download patterns by region or by country for articles matching keywords of interest. So as a small example from the data that we looked at, because I think it shows such an interesting pattern driven by events in the news, if we look at just articles with the keyword of Novichok, in terms of the number of documents published by country, you can see the UK, Russian Federation, and Czech Republic published the most, followed by the United States, which is actually relatively rare, as you saw in the previous slides. For most diseases, the United States publishes the most. The number of documents published per year is obviously very low until 2018, when we see a spike, and then even more articles published in 2019, mostly due to the use of non-proliferation, I think. And then we can look at the downloads by country, and what we're trying to see here is, are there increases or decreases in downloads by country that would indicate something other than basically this pattern. The number of downloads for any given publication in a year is often less than 50. So I told you we initially started this with Elsevier because Elsevier was interested in what kinds of reports they could provide to their authors and whether this would encourage authors to kind of stick with an Elsevier journal or encourage libraries to keep Elsevier journals. They decided that this would not be helpful for that because there are so few downloads, and they thought that that authors wouldn't want to see a report that says, your article was downloaded 20 times. I feel like that's very demoralizing. So it ended up being more useful, I think, to me than to Elsevier. But this is the kind of thing where, so this is a sort of point-in-time sample for this one keyword, and I think it will be interesting to compare this to other keyword trends, and also to continue monitoring over time, with the exception of the kind of COVID break. But so that's part of what we're refining this model to do now, is to be able to project and say, based on this historical data and based on a graph like this, but for other keywords, what sorts of download patterns do we expect? And then also, are there spikes in downloads before a spike in publications or before some kind of event, so that we can say, why was there this interest that happened for this? We can also track and did track, the downloads by IP address, which I guess could get us to by individual institution or even by individual. The graphs end up being more complicated, but that's something else that we're also looking at tracking, and that's something where I think the LinkedIn data will be helpful, or getting more language expertise would be helpful, because then I can start looking at individuals and individual patterns of downloads. And I will say, to my cybersecurity colleagues in privacy, I'm sure this is something that is a little bit discomforting, because you don't want, I think, your reading patterns to be tracked. And if I were giving this talk, within the cybersecurity context, I would say that this is a sort of bad privacy progression, or something, not a privacy violation per se, but something where protections around, what you're reading at the library, for example, have historically protected against. But it is data that's provided, particularly for open source journals, but actually for all Elsevier journals. I think it's interesting to track. Part of what drove this, and this will be familiar to all of you who worked with Ray, is I initially wanted to look at this because I'm interested in building a model in terms of how we recognize non-compliance. So Sergei Popov, after he defected and was giving interviews on both Vector and Biopreparat, the Soviet Union bioweapons programs, criticized US intelligence for not recognizing that Soviet scientists were being less communicative and less prolific when they began bioweapons work. And so in an interview with Homeland Security in 2000, with Homeland Defense, he was asked, what about your former colleagues? Do you believe that any of this work you discussed is still going on? So this was even after he defected. They were saying, do you believe that Russia is still conducting this work? He says, yeah, I'm pretty sure they are. I don't have any direct evidence, but recently I've begun looking up what my former colleagues have published. All I found were a few lousy papers. This suggests they are currently working on something that they cannot publish, and that's a good indication that the program is still functioning. Those papers are just cover stories. Yes, that's also allowed to publish. So having read this in a book that I think Michael Nacht assigned us in a course I took with him at Berkeley, I was just really interested in the idea that this is something that we can track. This is something that could be going on, and that also, even aside from indicators that people are not publishing this kind of negative information, we can look at both how people are publishing directly before a drop-off, or where people are, in the case of downloads, where people are reading a lot of articles but not publishing subsequently. Usually you download papers that are related to what you're working on. Usually you would see kind of maybe a lot of downloads and then a publication. If we don't see that pattern, maybe that's an indication of something. An indication of something we could follow up on later. Another way of looking at what can we track, so this is now just the publications by country, again stocked at right before 2019. Selected countries of interest to BWC verification. So this is kind of trying to, again, feed into the model baseline research activity that in some cases can demonstrate funding trends. And so this is based on author affiliation. And then we can also track by keyword. So by country, by keyword, number of publications by year, indexed by pathogen of concern. So this can indicate local centers of expertise. maybe changes in funding, maybe changes in staffing. If we look particularly with Russian author affiliations, we can look at keywords that are unrelated to dual-use pathogens and keywords that are related to conditions related to pathogens of concern. Some of this, I want to go back to Scopus and see whether there are changes in the way that they indexed papers to see if these are kind of real signals or not. Some of them have similar profiles. If you look at diabetes, tuberculosis, mycobacteria, we don't really need to know, I guess, why. There was an increase in research in 2015 as long as it's consistent across all of these keywords. We could say that could be due to new research funding, the opening of research institutes, or perhaps difference in the way that Scopus tracked this. But then there are some interesting gaps. So the kind of the dip that you see in the graph of Russian publications overall is reflected and even kind of exacerbated when we look at radiation, which I think would be interesting to look more into in terms of why there's kind of this dip in publications on radiation that immediately recovered maybe 10 or so years later. So these kinds of indicators are things that I want to be able to track over time and then try and trace back to things like funding, like institutional openings. The caveat, of course, to all of these methods is that whenever we're trying to do kind of infodemiology, we have to think about not overfitting a model. So Google Flu Trends tried to predict flu and ended up making several errors in terms of overfitting their model, testing on training sets, and then ended up kind of drastically over-predicting the intensity of the 2013 flu season, which was kind of right after they had stopped using all of their training data. So the reason why they wanted to do this is that CDC's flu surveillance is slow. There can be 12 days from the start of the morbidity and mortality weekly report week, 5 days from the end of the week. We might want to predict 1 or 2 days out if we can. So the idea was to use a real-time estimate of Google searches, get kind of historical ground truth as training data, and find search queries that correlate well. Some of the things they didn't take into account that we'd want to take into account in our model are correlated to under or over reporting, overfitting of the model. In Google, for example, they didn't account for changes to the Google algorithm itself, which I think is why it's important to work with ScienceDirect, work with Scopus to understand how they're indexing articles. So that is kind of, that project is in process. So I've shown you, some of the kind of initial data that we're feeding into the model, and we're hoping to have a model that we can then follow over time, track indicators of where publications may differ. The second project I'll go into on health facilities, CyberBio assessments is much more kind of procedural and recommendation based. And my idea here is that cyber risk assessment could be an additional confidence building measure. The difficulties with this is, as I said, the confidence building measures are not particularly onerous, and yet not very many people report on them. And so do we want to really add another one? I would recommend that we do, just because I think the opportunity for a leak or an incident that is actually the result of a hack, but that doesn't seem as though it's the result of a hack, particularly if you think about something like Russia's accusations at facilities in Georgia, where Georgia then invited site visits and the US decided that there was no illicit activity going on. But you might imagine that the next step for Russia could be then to initiate a cyber attack against that facility. If this leads to an outbreak, then now Georgia is saying, this wasn't us, we were hacked. And that's kind of a common story that you see here, where companies claim to be hacked, and then sometimes it was a hack, sometimes it was their own kind of lax security. So the case study I'm using here is some consulting work that I did. The case study is from Kaiser Permanente. I think it applies to any sort of large health or biotech facility that either stores sensitive data or has biomedical devices. The general process that I'm recommending is an identification of all networked devices, which doesn't have to involve a site visit. It can be done remotely. And then some risk categorization, securing and remediating devices by that risk categorization, and then developing an ongoing compliance plan. So the general work stream here is to look at all sort of edge devices on the network, so devices that are not computers, that are not workstations, but devices that are connected, but not subject to kind of an IT protocol. So these include kind of facilities, operational devices, and biomedical devices. Categorize and classify them by risk, just basically by tracking the CVEs and making sure that where patches are available, they're applied. Securing and remediating devices by category as capability allows, essentially, and then developing capability to ensure ongoing device compliance. So some of the methods for estimating the total number of devices, we can do a network ping sample analysis, use industry benchmarks for the percentage breakdown of devices, data analytics like machine learning on the network to identify devices that connect at a regular time frame, pilot physical account across a few sites if site visits are allowed, or extrapolating those across an entire organization, or location inventory analysis, so self-reported device inventories. Categorize the devices into 3 major groups, so biomedical devices that may store or record private information and are used to diagnose, prevent, or treat, facilities operations devices that are used to monitor and maintain functionality of operations in the facility, and then other edge devices, so other uncontrolled IT equipment that does connect to the network even if sporadically. Within those devices, we usually find that biomed devices are the largest, about half, usually the devices identified fall into that group. A typical device would be like a clinical device or an imaging device that could be attached to the internal IT network as well as to a separate clinical network. And then facility operations devices, so HVAC systems, cameras, building security devices, usually don't contain confidential data but can be under remediated, are usually kind of the last priority in terms of applying patches, and then other sort of workstations, servers, traditional IT devices. The risk profile is defined collectively by the device attributes, access to information, ability to control information or to control other devices, and potential to cause significant performance risk. And then the device risk profile levels, either the device is not connected, so if compromised it won't affect the network, we usually don't do anything to those kinds of devices. Low risk, so when compromised it threatens efficiency or threatens individual records, individual sources of private information. Medium risk and high risk. And so what we do there then is that we sort the devices that we've found into these 3 categories and then into these different device risk levels. This example from categories kind of where we sorted different devices that we found on their system. And then decide, are we going to de-scope the device and just catalog it, indicate that the device is there? Are we going to segment it from the network in order to protect it? Or are we going to remediate it using different software, you're applying patches on each segment? And then what is kind of the institutional maturity level we want to have in terms of cybersecurity within these 3 categories? So from these devices, some of the classification criteria we found here involve, can you store private information on this? Does the device support secure access? Does the device have access to critical data? Can the device boot from uncontrolled media? Is the device in control of patient lives? Is it at the center of a business function? And does the manufacturer recommend the devices being high risk? So these are kind of some questions that we would ask in terms of defining the classification of any given device. Here's a sample set of devices that fall into each category. In most environments, only a small number of devices will be classified as high risk, so kind of the minimal recommendation would be to focus on those devices first and remediating those first. And then the primary protection levers here fall into those 3 categories. So if segmentation is not possible and the breach won't impact any critical function, we can de-scope it. If segmentation is not possible but the device contains significant private information, then we want to push it to remediate and so on. And there is residual risk even using these remediation levers. So the remediation levers will leave behind residual risk by category. So for example, a device may still have inappropriate access levels for some users. The device may use an outdated or unpatched operating system, which wouldn't be covered by applying patches for the CVE search. Or there may be out-of-date patches or newly identified security vulnerabilities for which there are not patches available. And then there are also behavioral aspects to remediation that we want to keep in mind. Preventing unauthorized physical access, storing data in secure locations, keeping devices offline when not used, and then other recommendations that fall within those categories. Facilities devices that are not directly involved in patient care are common targets. And so these are just some examples of devices that you might find in a production facility or in a pharmaceutical facility as well as in a healthcare facility. And then some very critical CVEs that apply to each of these. So some of these were actually found kind of during this process of device remediation. And all of these that I'm listing here were unpatched in the devices on which we found them at the time. So these include things like chilling and sterilization systems, or you could imagine that if your sterilization system is not operating correctly, you may think that your devices are sterilized and they're not. That could lead to an outbreak. If your power supply can be remotely turned off, if your access control panels or servers are able to be breached, if your refrigeration monitors can be hacked and turned off, any of these kinds of things you could think of as leading to incidents that we would rather not have happen, particularly at the facilities that we're reporting on in CVMs. So the general thing that we're doing here is segmenting the devices from the network, so kind of isolating them from use and then patching them. The example I'm giving here is from that vulnerability from sterilization equipment, because as it turns out, these two manufacturers of sterilization equipment comprise 57% of EU sales by volume. So if you have one vulnerability in the microcontroller that controls both of these two devices of sterilizers, you can imagine that one vulnerability, if you're able to access this across multiple systems, now you have sterilization procedures across multiple hospitals or across multiple facilities. I bring this up to kind of make it seem more important than it may already seem to apply these patches to something like a facilities operations device. These devices pose a significant challenge because they have some unique characteristics where remediation is often dependent on manufacturer recommendations. There's usually no kind of off-the-shelf security software, and there's no McAfee for sterilizers. Typical devices only have enough system capacity to run basic software, and the device risk profiles depend on sort of how smart the device is, but that will also, smarter devices also mean that you have less ability to apply patches or less ability to change their operating systems. So this was similar, the previous slides I showed you for biomedical devices, these for facilities operations devices, similar questions were asked, similar lists of devices, similar protection layers, residual risk, behavioral remediation levers for these as well. So this kind of framework is what I'm proposing would be added as another confidence building measure to provide an audit of devices at secure facilities and recommend sort of cybersecurity maintenance and remediation activities. Seems kind of relatively basic. I guarantee it's not being done because it wasn't being done at Kaiser, it wasn't being done at Johnson & Johnson. So I think this would be kind of a way to encourage this to happen, particularly at facilities where it could be important. And then the third project, I will kind of switch gears again. This is a proposal for secure multi-party computation to enable better sharing of sequence data. So the problem here is synthetic DNA providers have to identify techniques used by creative adversaries to sneak banned sequences into their orders. And they do this using an evolving sort of series of programs. So there's one called FungiCat, developed by DoD, ThreatSeek, which Twist Biosciences is currently using. And all of the coordination and implementation of this is voluntary. So not only which program is being used, but kind of the use of a program at all, as well as I said, the sharing of results of this program from company to company. My current position involves a lot of kind of California policy. So one of the bills that I was advocating for before it got vetoed was this California Assembly Bill 70, which would have made it mandatory for genetic synthesis providers, at least in California, to share this data and to participate in this consortium. The two problems that the governor identified with the bill when he vetoed it, one was that the funding for kind of establishing the consortium would come from the companies themselves, but the companies would then have to pay into the system before being able to make use of the system, which I think is a change that we can fix in subsequent iterations of this bill. But the second problem identified in the statement on why he vetoed it was he said that California should not be making the first move on this. This is something where there should be federal legislation on this, which I sort of agree with and sort of don't, because California has moved first on consumer data privacy legislation, and that actually has spurred a lot of federal action. And I don't think that California shouldn't move first if it identifies a need for this. So I'm optimistic about being able to rework this bill in such a way that it passes his approval. But going back to, okay, so what does a synthesis provider do, and kind of what are these algorithms looking for? So they're searching for blast alignment scores of the sequence to known sequences based on a 200 base pair window. And I have an asterisk there because earlier programs used a sequential window. Newer programs are using a sliding window. That was one of the kind of vulnerabilities that people suggested could be taken advantage of, which is that if you're looking at subsequent windows, you could have something that's kind of in the middle that matches a bad sequence, but if it doesn't, if you know how the window is going to be evaluated, you wouldn't necessarily find that. It used to be that you were only looking for restrictions on cut sites. Now, of course, with CRISPR and Cas9, we're looking for Cas9 cut sites, which expands the population of sequences we're looking for. There's always the possibility of assembly through very small orders, so Gibson assembly. In that case, those pieces would be too small to use these screening algorithms on. And it could be, the smallest kind of toxin is a 90 base pair toxin. So if you're using assembly with very small sequences, you may just be missing scanning for assembly that ends up in something larger that you want to look for. Ordering from multiple providers, as I mentioned, because the companies don't have to report what is being ordered amongst themselves, you could order small pieces of a sequence from multiple providers. And then inserting obfuscatory sequences. So there are proposals for ways that you can take a gene that is maybe the second alignment score for something that is for a bad pathogen, or for a gene from a pathogen, take something that is from an organism that is not prohibited but matches up very closely, and then insert it in such a way that for every sort of 200 base pair window, the closest match is from that gene, but you're able to then remove that with a Cas9 cut site. So if you think about it, these are all things that synthesis providers have to keep in mind, either when they're developing their own program, when they're evaluating what program to use, or when they're screening and then reporting to other providers. So my thought was, this is a lot to ask of an individual synthesis company. What if we let anyone query the sequences? Sharing of sequence data is voluntary, in part because synthesis orders are private. There might be trade secrets for new biotech products. But this means that anyone with a new algorithm, or anyone with an idea of how a creative adversary could circumvent an algorithm, has to work through a synthesis provider to have them test this algorithm, and can't test on real data. So the thought is, can we share the actual synthesis orders via some kind of encrypted database that anyone can then query and compute on? So this brings up the idea then of using secure multi-party computation for this kind of database. So the idea is that we have maybe these synthesis providers, or maybe just labs and researchers that all want to share some private information, which could be sequences and then a threat pattern or a model that they want to compare against. And they want to share some public information, which is the similarity score that they're computing. So they have a threat model. Maybe it involves, if you use this Cas9 cut site and this Cas9 cut site, and you put, this piece of DNA, this sequence in here, this would allow you to create this malicious sequence. And they want to compare that against all of the sequencing orders to see, well, has anyone done this? Or is there any sequence that matches this pattern? So at the end of this computation, where they each want to compare their private sequence, their private threat model, against sequences that have been provided to each of the other parties, at the end, each party learns the similarity score for their threat pattern against all of the sequences in all of the others' databases. And then the privacy guarantee that we want is that no party learns anything extra about the other party's inputs. So they only learn that similarity score. They don't learn what the sequences actually are. So the simple version of this we can use is two-party computation. And this is mostly a solved problem, something called Yao's circuits. You can express your function as a Boolean circuit. So for sequence comparison, matching at each base pair is either true or false. So if we think of a sequence where we have 4 base pairs, the string that we want to use then is going to be twice that long, because we'll have to encode basically 4 items in a Boolean 1 or 0. And we'll just compare then at each step whether the sequence matches your kind of pattern sequence or not. And so this can be used to evaluate circuits, Boolean circuits, very efficiently, 1.2 gates per microsecond usually. And the way this works is that synthesis provider A prepares a garbled version of their circuit or their Boolean string. Synthesis provider B uses an oblivious transfer protocol to transmit a garbled version of their input, so their kind of pattern to match, and then synthesis provider B can then learn the output of the garbled circuit. There's a really nice way that, so this is work that I've done with Dan Bonnet, who's a cryptographer at Stanford who I've worked with, and he has this really nice diagram here that explains how two-party Yao circuits work. I don't have to walk through this with you now, I guess, based on time, but I think it's a very elegant way to think of how this works in terms of, you have a key, someone else prepares a bunch of different boxes. Inside of other boxes, you can only open the outer box with your key. If they then open the inner box with their key, then you're able to see whether you were right or not. Whether your Alice's true matches Bob's true, and so on. Essentially, I guess if you believe this, or if you read Yao's paper, this allows us to do that circuit evaluation quickly and efficiently. So that's with two parties. So this is two-party computation, two-party circuits. But we can actually extend this to multiple parties. It's certainly feasible. It requires more communication. So general kind of multi-party computation suffices to evaluate arbitrary functions with many parties. The limitation is that it requires many rounds of communication, large bandwidth, and it can be difficult to coordinate these interactions among many parties, particularly if there are sort of time delays. So what we could use instead is homomorphic encryption, which would require us to use only a few rounds of interaction. And with homomorphic encryption, we can compute on arithmetic circuits instead of Boolean circuits. So the way this works, homomorphic encryption essentially is regular encryption, but with arbitrary computation. So if we have two encrypted messages, M1 and M2, and then we have an optional evaluation key, we use an evaluation function to get out a third sequence, and then when we decrypt the evaluation function evaluated on the evaluation key, message 1 and message 2, or rather, C1 and C2, which are the encrypted versions of message 1 and message 2, what we get out is essentially the evaluation function evaluated on message 1 and message 2. So there are many homomorphic encryption schemes that we know of. Algol encryption is homomorphic across multiplication. Pi-A encryption is homomorphic across addition. BGN encryption, you can do one multiplication function, but you can do as many addition functions as you want. But fully homomorphic encryption that allows you to do addition and multiplication would allow you to compute any arbitrary Boolean circuit. So fully homomorphic encryption exists. It is very computation intensive. So is there a way that we can kind of simplify then the computational requests that we're making? So this is the idea of having something like a secure sequence cloud. So HHS or some trusted identity publishes a public key. Each of the sequence providers report their encrypted sequences and potential threat models, and then they're able to do this computation along with any of these researchers, for example, who want to compute in this cloud, and then only HHS or this trusted identity would be able to view any of the information that's in the cloud. So what we do is first, so HHS publishes a public key. Each of the sequence providers or researchers upload their encrypted values. We can compute the model, which is essentially comparing the sequences against the threat model. We can compute that homomorphically, and then we can decrypt to obtain the output of the model. And by doing this centrally, we save a little bit on computation. An interesting thing here too, though, is that if we have an adversary in the cloud, a passive adversary sitting in the cloud would not see any of the client data, and HHS only receives the results of the output model, not the individual data points, and parties only need to communicate with the cloud. And do public key encryption, that means that the provider of the public key for the cloud is the only one that we could see as a potential adversary. So we only have to essentially trust one entity. We could also, though, do hybrid homomorphic encryption. And so this is more efficient, but allows us to still do the kinds of arbitrary computations that we want. And the way that we do this is we encrypt an AES key using homomorphic encryption, which is sort of computationally expensive. And then we encrypt the data using AES, which is not homomorphic, but that's sort of computationally cheap. And then we can homomorphically evaluate the AES decryption circuit. So these two clouds are the same. I just kind of expanded the clouds. You can see all of this computation is happening in the cloud. So we have an AES key, and we've used that to encrypt this message M. And then we encrypt the AES encryption of the message, homomorphically evaluate the AES decryption with the encryption of the private key K. That leaves us with the homomorphic encryption of the private key M, and then we can homomorphically evaluate our function on that and get the equivalent of the homomorphically encrypted function on M. So the reason why I'm proposing that we use hybrid homomorphic encryption for this kind of process for computing threat model evaluation against sequences is that fully homomorphic encryption exists. Microsoft, UD, IBM, and others have publicly available fully homomorphic encryption implementations, but it's computationally expensive or time expensive. And the reason for that is that these models these implementations allow an arbitrary number of computations on the same ciphertext. My thought, though, is that we don't need to allow an arbitrary number of computations on the same ciphertext if we're only comparing sequences. And so when we implement something like this on a 64-core machine that Dan Bonet kindly allows me to run things on, with 6 gigabytes of memory, we can compare 1 million base pair comparisons in 1 minute. which is not fantastic, but I think would be usable, particularly if this is something where we're running it every day and not every minute, every second. So this kind of is my proposal, which I have not run by any synthesis companies yet. This is kind of my proposal for how to get around this sort of information sharing problem where we want synthesis companies to be able to communicate with each other, and we want also, I think, we want any researcher to be able to propose a threat model and see, are there any sequences that synthesis companies are being asked to provide that match this threat model, rather than kind of working through adding this threat model to the DoD algorithm that then they have to convince Twist Biosciences to use, and then they evaluate on their sequences. So that's sort of the idea of we could preserve privacy via this encryption cloud and still allow computation across all sequences.